and welcome to Claret Bio's quarterly webinar series, the Series Talk. Our goal for this webinar is to provide an opportunity for our customers and collaborators to share their work with a wide range of audience members and engage in discussions amongst other academics in the field. At Claret, we specialize in the development of NGS library prep and analysis tools that empower researchers to get the best data from even their most challenging samples. A few housekeeping tips for today as we progress through today's presentation. Please submit your questions via the Q&A box down in the corner. At the end, we will dedicate the last 10 minutes of today's webinar to addressing any questions. Now, without further ado, I'm delighted to introduce our guest speaker, Dr. Katie Miller. Katie Miller is a principal investigator in the Institute of Genomic Medicine at Nationwide Children's Hospital and an assistant professor of pediatrics at the Ohio State University College of Medicine. Dr. Miller leads a research lab whose efforts focus on the characterization of pediatric disease-involved tissues using state-of-the-art technologies, including transcriptome sequencing, single-cell RNA sequencing, and spatial multiplex protein profiling. Dr. Miller maintains active funded collaborations with neurosurgeons, surgical and clinical oncologists, and basic science researchers with a primary focus on studying pediatric cancer and epilepsy. In addition, she serves as the director of Genomic Services Laboratory at Nationwide Children's Hospital, staying on the forefront of genomic technology advancements to enhance the Institute's technological capabilities and services offered to all investigators at Nationwide Children's Hospital. We're glad to have you here today. We look forward to your presentation on post-zygotic rescue of meiotic errors causes brain mosaicism in focal epilepsy. Thanks.
Thank you. And like me, I'm very excited to show the work we've been doing at our Institute for Genomic Medicine at Nationwide Children's Hospital. My talk is going to focus on pediatric epilepsy, and I'll show you some of our ongoing work and then some really exciting recent findings that we published. I want to first kind of introduce the topic of somatic mosaicism and why it matters when we study epilepsy. So epilepsy is a pretty common disease actually in children, and a portion of them, maybe like a quarter to 1/3 of them, don't actually respond to medication. Right, so common medications you might think of to treat seizures, they don't respond to. And so for a small subset of these patients, depending on a lot of things, actually surgical resection is a second-line treatment option for them. So they actually identify through various mechanisms where the seizure activity is occurring in the brain, and they resect that. The efficacy of these kinds of surgeries is variable. But it is sometimes curative for these children. And as researchers, it gives us a very good biobank, if you will, to study these tissues that have been resected from these children. So a little bit about somatic mosaicism, if you're not familiar with the topic. This is basically a phenomenon in which cells, as they divide, might acquire a mutation or a genomic variant in the cell. And they retain that, right? It might offer a selective advantage to the cell or something. So they retain that mutation or variant, and then it's in all of the subsequent daughter populations. So it's somatic mosaicism because it's often, at least in epilepsy, very localized in the brain, and it often correlates with where the seizure activity is occurring. But this in general is like a phenomenon that happens even in healthy people, right? It just depends on what gene the mutation occurs in and the timing of the mutation, what cell types it's affecting, et cetera. So in studying pediatric refractory epilepsy, if you will, a lot of studies over the years, right, have contributed to the somatic mosaicism kind of informing the pathology and vice versa. And what I mean by that is now I think we're to a point where when pathologists diagnose these, right, they always get the tissue first, right? The clinical analyses come first. So when a patient has a resection, the tissue is sent to clinical labs and they do pathologic diagnosis. And basically this is from a paper published in Brain last year, but if you can see on the bottom part, it's kind of putting these patients, right? Each column is a patient, and it's putting them into groups of what their pathologic diagnosis was. And then you can see in the rows, they have different genes labeled, and you can see there's sometimes stronger than not correlations with the genes that are mutated and what the pathologist is seeing, basically. Right. And this is important, like long term, because if we think about the pathologist looking at the tissue, they can think, hey, this looks like a FCD, which is focal cortical dysplasia, by the way. There's different types, but they can like actually see what's going on and then think, oh, this might be associated with a certain gene mutation, which maybe someday we'll have targeted therapies for, right? That's like a long-term obvious goal. But we study a lot of these patients in our institution, and I just noted some case reports that we've published on here, which basically match these findings, right? The tissue had a mutation in PTEN, for example, and it was very similar histology and pathology to what other groups have reported. So our approach at Nationwide Children's Hospital starts with the patients. We have an IRB-approved research study to basically consent these patients and families, if they wish, to enroll in a research study where we can actually obtain that surgically resected tissue. And so this is we published this work, I'll talk about it in the next few slides, 2 years ago in Epilepsy, but basically what we're routinely doing is collecting blood and collecting resected brain tissue, however much we can get. And then we're performing exome sequencing and also RNA sequencing, which I won't talk about today, but the exome sequencing paired with blood is really to help identify brain-restricted variants, right? So these are not in the patient's constitutional germline. These are restricted to the brain and likely associated with disease if we identify them. So we're targeting like 250x for our exome, and then we apply a bunch of filters and thresholds that I show to the right just to help us narrow down any variants that might be actually disease-causing. And one thing that's really helped us do this is because there's a lot of publications now about this type of work, we've curated a gene list that are genes that are commonly seen in epilepsy. So our first pass is like looking at that list to see if there are any obvious hits. Yeah, so this was the publication I was just talking about. So we published the first 50 patients in our study in Epilepsy Journal. And again, the FCD is focal cortical dysplasia. So there's 3 types of that that we know of. And then something called LEAT, which is long-term epilepsy-associated tumors. So these were a little bit unique in that they had brain tumors and epilepsy. So just showing the distribution of the patients and then showing the common genes and some of the mutations that we found in this box. And then the panel E is pretty interesting because we have the opportunity sometimes to get multiple tissues from these patients. So this can be different regions of the brain. Some of them actually get, I was really surprised to learn this, but some of them actually get like pretty large parts of their brain removed. It's actually amazing that they can recover from this type of surgery, but we can actually get multiple parts of the brain or the same part, but different tissues. And we just noted in panel E that you can see for the tissues, by the way, we detected in about 50% of these tissues mosaic variants. So not every tissue that we study has an obvious disease-causing variant, but based on our analysis and the knowledge that we have, half of our patients had a mosaic finding. And what we saw back to panel E was a range of allele fractions, right? So we might see this mutation at an allele frequency or fraction of like 5% in one tissue, but it might be like 30% allele frequency in another tissue. And that's important because we can start to correlate that, right? We have multiple tissues, and we wanted to start to correlate that with some clinical findings for these patients. So here I'm showing a case report that we published. This patient was a child with epilepsy. We studied their brain tissue from surgical resection, and they had a mutation in a gene called SLC35A2, which is very commonly known to be associated with this type of epilepsy. And basically what I'm showing in this graph is that we had, you can see it's not in the blood. And then we had different tissues, all from different parts of the brain. And we applied 3 different methods. The blue bar is showing our initial exome. And then we applied 2 more like targeted methods just to verify, like, yeah, this variant is real. Because some of them are pretty low frequency and they're kind of at the border of like, do we think this is real based on our initial exome? So we've gone back and done like deep targeted sequencing to verify that these are actually real and not just noise. But anyway, you can see a range of frequencies, the highest one being in the hippocampus at about 30%, right? So this means 60% of the cells in that tissue that we studied have this mutation, but in other tissues, it was much lower. So in this publication, we were able to correlate those findings that I just showed you with some clinical findings. So this was in collaboration with a neurologist here, Adam Ostendorf, because I know nothing about these EEGs or signals that they're reading, but we basically asked Adam if he could like somehow quantify this type of reading. And in the table, basically, what I'm showing is the triangle gradient that I show is basically, it goes along with the bottom row here that's showing the VAF or variant allele frequency of the SLC35A2 mutation. So from highest to lowest in the tissues. And then the 2 primary things that Adam helped us do were quantify spikes per minute, which is the second row, and then just give a descriptor, yes or no, of seizure onset in that area of the brain, right? So before surgery, these kids, they're hooked up to a lot of grids intracranially, and they basically follow them for a few days to see where is the seizure occurring, where is the onset, and the quantification would be spikes per minute. So we saw a loose correlation with basically the tissues that had a higher allele frequency of the mutation had like a worse clinical finding, if you will, in terms of the seizure activity, like the frequency and the onset of the seizure. So that's a broad highlight of what we've been doing so far. But applying just exome and RNA sequencing is not enough in terms of understanding the biology of the disease. And so there are a lot of questions that remain, but 2 of them would that I'm personally interested in my own research is what cell types harbor the somatic variants, right? We're not going to understand this from bulk exome sequencing. And also, how does the variant affect gene expression in those cells, right? So these tissues oftentimes, as I mentioned, have maybe only 5 to 10% of cells affected. So when we do any sort of bulk DNA or RNA extraction and study it, we're going to lose signal of what's actually occurring because of the mutation. So I just wanted to talk about one like technology that we are trying to optimize in our hands. So switching gears a little bit, but to single cell RNA sequencing, because we are doing a lot of that too. There are limitations with most technologies, if you're familiar with these, because they basically read the RNA from the 3' end. We're interested in RNA sequencing, but also understanding which of the single cells have the mutation that we're interested in, right? And so I put here this lollipop plot as an example, but these are all the disease-causing variants that have been identified in a gene called DEPDC5, reported in the ClinVar database. And you can see they are just like spread out throughout the gene, right? So if you're reading RNA from the 3' end, and usually it's only 90 base pairs, you're not going to ever get coverage. Well, by chance you might, but you're not usually going to get coverage of the variants out here. So genotyping cells from single cell RNA sequencing data is not ideal with most current technologies. So one thing we're working on with our technology development group here, led by Anthony Miller, is integrating long read sequencing with single cell, like traditional short read sequencing. And basically on the bottom plot, I'm just showing the traditional workflow. The top row, we do single cell gene expression. And then in the bottom row, we would take that cDNA generated that's already barcoded with single cell tags and do long read sequencing to get longer reads of the transcripts. And this will hopefully help us get reads of the variant that we care about. And I'm just going to show one. This is actually not the focus of the rest of my talk, but I'm just showing one example here where we had a sample. This was not epilepsy, but this was like a benign malformation in a child that we studied. And so basically what I'm showing is that we knew there was a C to T mutation in a gene that we cared about. And when we tried to genotype just from short read data, you can see on the left, only about 1% of cells had coverage for that variant so that we could genotype it. And when we coupled it with long read, we increased that to 15%. So this is ongoing work, but something we're very excited about. So for the remainder of my talk, actually, and the most exciting thing, I think, is a paper that we published late last year. And this focuses on meiotic errors, actually, that are a previously unrecognized, actually, cause of somatic mosaicism. Okay, so this, we had a nice cohort of 6 patients who all had the same finding. And I will say these 3 of them came from our institution, and another 3 came from actually different pediatric hospitals where we kind of crowdsourced, if you will, like kind of got the word out of these findings and were able to get other tissues. So all 6 of these patients had, if you look at their onset age, the third column is very young, right? So basically infantile onset of seizures, pretty severe. And the pathology image I'm showing is, they all had this finding, and it's called astrocytic eosinophilic inclusions. So I'm not a pathologist, but this is an H&E stain, and the arrows are pointing to these astrocytes that are have these eosinophilic inclusions. And so it's a very unique finding. It's apparently not something they see very often, or at all, actually, in these cases with epilepsy. But we had 6 patients that we wanted to study. We knew they all had these astrocytic inclusions. And we also knew from our profiling that I told you about that they all had a mosaic finding. And this was not like a point mutation. This was a gain of chromosome 1q. And so I'm showing 3 different figures at the bottom to basically show how we detected this gain and what it looks like. Right. So again, we got the brain tissue from all 6 patients and we coupled it with blood, did exome sequencing. And I'm only showing chromosome 1 here, but you can see there's like a blip on the Q arm, right, which is the one on your right, probably. So you can see that there was a gain in that, right? So that means relative to the blood, this patient has basically extra 1Q material in their brain. We also did FISH studies, so fluorescence and C2 hybridization. Here the orange is 1P, which is diploid as we would expect, and the pinkish color is the 1Q. And so you can see for this particular patient, they had 4 copies, right? So 2 extra copies of 1Q. And then the third thing we did was single cell DNA sequencing. And we were able to, from one patient, sequence about 880 cells. And we basically saw, in most of them, 70%, they were diploid as expected. But in 30% of them, they had 4 copies, right? So this is tetraploidy for chromosome 1Q. So we did some further analyses with single cell DNA sequencing, right? These single cell DNA sequencing actually is not, I don't think it's something a lot of companies at least are focusing on. They're primarily focusing on RNA, but we knew if we could just do whole genome sequencing on single cells, like we could get what we needed, because from our initial exome, we couldn't tell the exact number of copies. And that's why we did the FISH and the single cell DNA to understand like what is the absolute number of 1Q in these tissues. Excuse me. So you can see very clearly 2 things. One is the genome is very flat at the bottom, except for the 1Q. And this y-axis is on a log2 scale. So you can see there's 2 extra copies of the 1Q. So this is where things get very interesting, actually, because the way I think that I always thought about it, and I think most people think about somatic mosaicism, is that these are variants that are acquired at some point during development, right? And if you know anything about cancer genomics, it's very similar to that, right? At some point during development or in your life, you acquire a mutation in a cell. It's like in the wrong gene at the wrong time, if you will, and it causes disease. So we always think of this as post-zygotic mutations. That's a common term used for this. And there's like two different ways, I guess you could think about this, showing from this review paper. But one on the left here is that you might see the somatic mutation in all tissues, in like brain and non-brain tissues, but you don't see it in the parent samples if you have those to sequence. And so this would indicate like a de novo mutation that happened early in development, because it's like everywhere in the body. And then the second scenario on the right would be that you see a somatic mutation, but it's very localized or focused in a certain tissue. And again, you don't see it in the parent. But the data I'm going to show you very much contradicts this, because what we saw is that it wasn't something where they acquired this extra 1Q at some point in development. It was something where it was present from the beginning and it was lost in all the tissues except the brain. So the first thing that tipped us off to this was from our exome sequencing data, right? This was short read sequencing, but we essentially know the profile of the extra 1Q material. And initially, if we're thinking of it as like a post-zygotic event, right? I would think that it's just a duplication of their existing 1Q. Like they have chromosome 1Q and somehow it duplicates itself. Here, this was tetraploidy. And so you would expect that the SNP profile would match, but that's not what happened. So here I'm showing you for one of the patients, but we saw this in our cohort, is that these are all the variant, not all, these are a subset of the variants on chromosome 1Q. You can see for the multiple tissues that we studied, we had blood, we had tissue A, B, C, D, E, which was different portions of the brain. And you can see the allele frequencies that we detected mutations on the 1Q. But then in the last column, you can see the NOMAD population frequency. So NOMAD, if you're not familiar, is a database that has like tens of thousands of exomes and genomes. So we routinely look at these mutations just to see if they're in the population, right? Because you would think if it's a very rare disease-causing event, you're not going to see it at 5% of these supposedly healthy people that are in the NOMAD database. So we saw that a lot of these mutations were actually very common. And so we actually suspected that this chromosome 1Q was actually inherited, right? These are not just like rare events that are happening all across chromosome 1Q, like these somatic mutations. This is actually like this patient and the patients in our cohort had a completely different chromosome 1Q than what was in their blood, like what was in their constitutional germline. And actually when, I should talk about this first. Yeah, so we wanted to know, so we hypothesized that it was inherited. And we wanted to do deep targeted sequencing of just the chromosome 1Q variants that I just showed you. And this was really to see if there was evidence of mosaicism in the blood tissue that we'd already sequenced with exome, but it's pretty low coverage. So we wanted to get like really high depth sequencing to see, is there like any evidence of mosaicism in the blood? And we also wanted to sequence the parental samples, which we were able to collect, to see if they had evidence of this 1Q. But this was kind of challenging because we had already used a lot of the brain tissue and a lot of the DNA for the assays that I'm showing you here. Like we threw everything we could at this study. We were so excited. So we knew there was a need to apply an approach that would allow for basically a low input prep to do this NGS study that we wanted to do. And luckily, we in our institute, primarily in our technology development group in our clinical lab, have already been thinking about this because not for my research study, but in general, we have a lot of samples that come into our clinical lab that just don't meet the threshold for like the optimized exome or genome sequencing assays that we have, right? So a lot of kits will say you can go down to a certain amount, but we don't tend to do that unless we've optimized it in our own hands. And we had already been working with the Claret Seriously kit to do that, right? So one of the main things, just a plug for our clinical lab, is that we are part of the Molecular Characterization Initiative, which basically sequences all comers, all tumors from the Children's Oncology Group. And they noticed, I'm not part of the clinical lab, but they noticed very early on a need for a kit that could prep low quality or low quantity amounts of DNA. So as a researcher, I knew this was already available to me in our institution. So it was obvious that I wanted to do this follow-up study, but I didn't have much DNA. And just a plug, if I assume a lot of people are familiar with this kit, but the thing we really liked about it was the lower input amount. And it's prepping nicked DNA and it's prepping single-stranded, which the other kit that we used does not. So these samples are very precious. These are patient samples. We'll never get them again from the same patient probably. So we wanted to make the best use that we could of the sample that we had. So the way that we did this follow-up, again, we wanted to target just the 1Q variants in the tissues. So we designed a hybridization capture panel of basically probes that just span 1Q, right? So we wanted to only sequence chromosome 1Q so that all the sequencing could go to that and we could get really high depth to really understand if there's any level of mosaicism in the other tissues besides brain. So the graph on the right, I'm just showing we prepared the, nice little typo, we prepared the Claret library and then we used our capture panel to capture just chromosome 1Q. And for each patient when it was available, we tried to sequence blood, buccal tissue or cheek swab, the brain tissue that was affected, and then we had maternal and paternal blood. And then we always sequence a human reference sample because with these targeted sequencing assays, we're going very deep, like sometimes thousands of X coverage, and there's always going to be noise. So we use this as a negative control, if you will. So we prepped using the Seriously NanoPlus kit, and we aimed for 1500X coverage per variant that we cared about. And again, the two primary aims of this like follow-up experiment, if you will, is was the gain present in other tissues besides brain, and are the variants that represent that extra 1Q material present in the mother or the father or neither. So what I can tell you is that this indeed was detected in mother for all of our patients, and therefore it was inherited from the mother. So what I'm showing on the left is just a representative graph of one patient, right? We had 6 in our study. This is just one. But you can see at 0, it might be hard to see with this scale, but I can tell you all of these symbols at the bottom, which represent the patient blood, the patient buccal, and the father blood, did not have evidence of these 1Q variants. The brain, as we already knew from exome, had mosaic levels. So this patient in particular had around 20% allele frequency. And then in mom, we saw every single variant at heterozygous level, right? And so what this means is that this extra 1Q actually represents the mom's other 1Q, right? That she didn't perhaps pass on to all of the cells in the patient's body, but it's her other 1Q, right? So we have 2 chromosome 1s, right? One of them we think is passed on in the egg and the other one is not. But in this case, both of them were actually passed on. But I can tell you why we think it's only in the brain for one of them. And then on the right, just another depiction of what we saw. So the patient brain is at the top here. They had mosaic levels of these nucleotides, and they all matched perfectly with the mother blood. And I will say the mom was karyotypically normal. She didn't have other, some of the moms in the study had other children and they were karyotypically normal, which led us to the hypothesis that I'm going to tell you about. So we think the way this is happening is that there are 3 different events that are going on. The first 2 events I'm going to tell you about are pretty rare. So we got like a little bit of pushback from our peer reviewers about 2 very rare events occurring back to back. But to us, this was like a very unexpected finding. And to us, this was the most logical biological explanation that we could think of. So what we think is happening is that I'm just going to talk like left to right through this image. So we think that in meiosis 1, there's something called meiotic non-disjunction, which is a known phenomenon, right? Rare, but known, in which the chromosomes did not separate properly, right? So here I'm showing, just for simplicity, chromosome 1. So pink and green, each chromosome 1. We're diploid organisms, we have 2. So those did not separate properly, and both chromosome 1s ended up in the same oocyte. The second event that we think happened was something called centric misdivision, which occurred in meiosis 2. right? And this happens when they don't properly separate by the centromere. So instead of 1P and 1Q staying together and separating, it was the 1Qs stay together and the 1P arms stayed together. And this is called isocentric chromosome, actually. And so this resulted in an oocyte with chromosome 1, but an extra isochromosome 1Q. We believe it was fertilized with a monosomic sperm, which ended in this karyotype that I'm showing in the middle column here. And then the third thing that happened, which actually is a very common phenomenon, trisomy rescue, or in this case, tetrasomy rescue, is that cells can correct themselves. So if they have this type of event, they can correct themselves, right? And trisomy 1Q, by the way, is not compatible with life. So it makes sense that if this kind of event is compatible with life, it's at mosaic levels. So we can only study blood and buccal besides brain, but we don't actually know if it's in other tissues, but we presume not, because the only clinical phenotype this patient had was neuro-related. And we just don't think it would make sense to be compatible with life if it was in other tissues. So what we think is that the tetrasomy rescue occurred, but only in a portion of cells. And my hypothesis actually is that this extra 1Q provided actually a selective advantage for the cells, because we did a lot of reading on cancer, and a gain or amplification of chromosome 1Q is actually a very common finding in tumors, especially in brain tumors of glial origin. And so it makes sense that the 1Q perhaps provided a selective advantage for these cells to not rescue themselves. But in the end, ultimately, it caused disease. So that is our theory of why this is mosaic in the brain, which, remember, goes against, I think, the wide-standing theory that these events occur post-zygotically, right? So here we're showing this was actually an inherited alteration, but for this reason, or maybe some other reason that we don't know about, was restricted to the brain. So this is basically another way of showing our findings. So there was, after we published this in Nature Genetics, there was a News and Views follow-up by Joe Gleason and some people from his lab. And so this is basically another way of showing what I just hopefully explained somewhat simply. And basically is that the egg, again, upon fertilization, already had the extra 1Q material, but that was lost throughout development and but retained in the neural tissue. So the last few slides I have are on the single cell RNA sequencing that we did, right? So hopefully you remember I told you about that astrocytic inclusion thing that we saw, right? So we got very excited about the finding of the meiotic piece that I just told you about. But another major finding that we wanted to present to the community was this astrocytic association, basically, with the extra 1Q. And this kind of goes back to what I was saying about identifying the cell types that harbor these mutations or alterations in mosaic tissue. So for 5 of the patients, we had extra frozen tissue to do single nuclei RNA sequencing, and that I'm showing you on the left. So this was done by a graduate student, Nori, here at Ohio State. And basically, these are the cell types that we identified from all 5 patients. And we get very excited about analyzing this data because the cell types, as you can see, actually retain their phenotype pretty well. So it's very straightforward, I guess, to study these cell types and how they're affected by these alterations, right? So I'm thinking of tumors on the flip side, in that it's usually like a big blob and all the tumor cells kind of look the same. So these cell types in the brains that we study actually retain their phenotype pretty well. But the obvious question that we wanted to know was, where's the 1Q gain? And so we used a tool called Infer-CNV, and this basically takes expression from the cells and basically tries to infer copy number variation, or CNV. It's a little noisy, but we knew exactly what we were looking for, right? We knew these tissues had extra 1Q, and we didn't need great resolution because it was the entire Q arm of the largest chromosome in the body. So we knew very simply what we were looking for. So In fact, the infer CNV worked very well for us. And here on the right, this is a UMAP, by the way, if you're not familiar. Each dot represents a single cell that was sequenced. And on the right, we colored the cells in red if they had the 1Q gain based on our inference tool. And they're colored in gray if they don't have the 1Q gain. And then the bar plot, we summarized which cell types had the gain. And so you can see in all 5 patients that we studied, it was very clearly enriched in the astrocytes, which is very cool because they all had that very rare, unique finding of astrocytic inclusion. So perhaps the 1Q gain is causing these inclusions to be seen on the pathology. We did some follow-up studies just to kind of confirm this. So we sorted for astrocytes. So we kind of went backwards. We took the tissue and sorted for astrocytes and then measured if they had 1Q gain based on some of those SNPs that I told you about. And you can see in the bar plot that the PAX6 positive, which is what we use to sort the astrocytes, was indeed enriched for the 1Q gain. And you can do a lot of stuff with this. The paper's out if you want to read it. But obviously, one thing we wanted to know was how the 1Q gain alters gene expression, right? We expect to see upregulation of 1Q genes. That's obvious. But we wanted to know more about the biology and how this 1Q, the extra 1Q material is affecting. So we were able to perform differential expression, basically in cells with the gain and without the gain. You can compare their expression profiles and do pathway analyses and look for different signaling events. And so just when you can identify the single cells that have your variant or alteration of interest, it just opens up a lot to what you can do with the single cell data. So I will stop there. A lot of people to acknowledge for this particular publication and just the ongoing work that we have here in epilepsy.
Great. Thank you, Katie. I think there's one question from Tobin. When using long read sequencing for genotyping cells, what goes into your decision making for picking a specific platform, PacBio, Oxford, etc.?
Okay. Yeah, so you're talking about long read sequencing. I mean, for us, it was availability of the platform. So we already have the PacBio instrument here in our institution, and they'd actually already been working on a kit to basically streamline and make it very easy to use your single cell cDNA to go into that. So honestly, my initial thought is it was just availability.
Great. And if there's any questions, please feel free to add them to the Q&A chat or stick them in the chat. I have a question in terms of the astrocytes. So if there's opportunity for more samples, more patients to come in, again, do you think you would work backwards knowing what you know now? Or how would you kind of enroll new patients and what would the steps be?
Yeah, I think we definitely, you mean work backwards as in like start kind of with astrocytes? Yeah. I think definitely we would. And I think we would prioritize assays based on how much tissue we have, right? So I mentioned the, challenges with, like for here, for example, the single cell, for the 6th patient, I'm not showing them because we didn't have tissue left. So the challenges I talked about are real and the solutions I talked about are real. So I think we would definitely prioritize and getting fresh tissue would be amazing. But you can imagine working with the surgical team in the OR is just like a lot of logistics to get fresh tissue, because one thing we really wanted to do was karyotyping of the cells, like a traditional karyotype. And we tried that from cells that we hoped we had preserved, but it just wasn't possible. So definitely getting fresh tissue would be a priority of mine.
We have another question. Does single cell RNA-seq suggest that in brain, all cell types except for astrocytes and some neurons reverted the Q1 gain? Yeah, basically, you can see in the bar plot here, but it was more pronounced in some tissues, but excuse me, it was for the most part in the astrocytes and some neuronal populations, right? So we didn't see it
in microglia. Actually, we didn't see it in microglia, and we actually had, I mentioned we had multiple tissues from some of these patients, and there were some tissues that didn't have the 1Q gain at all. And then some tissues where it was only in astrocytes. So it's very like tricky to think about how that might happen, but that was our observation from this data. And we didn't see it in microglia, like I said, which is actually how we got this Infer CNV tool to work so well, is that we used the microglial cells as a comparator, saying like these basically don't have the 1Q gain, and compare it to the rest of the cells and predict where it might be, if that makes sense. Because if you have to use a comparator when you run this tool, and if it's not from the same patient, it gets really noisy.
And then Barsha has a question. Did you look at the indication of mosaicism in Q1 or other chromosomes in other tissues of siblings?
Oh, siblings from the patient or from the. We didn't have. Let me just read again. Did you look at indication? Yeah, we didn't have. So we had paternal samples for this study, but not siblings. But we did go through the family history and the chart notes, and there wasn't any indication of disease or like similar phenotypes in the siblings or in either parent. And then, as I mentioned, the phenotypes for these affected children were really restricted to the brain. So in terms of other tissues in the patient, we didn't have those other than the blood and the buccal. But as I mentioned, we don't think it would be present just given the presentation of the kids.
Thank you, Katie, for answering those questions. And with that, we will conclude this presentation. Please join me in thanking Dr. Katie Miller for presenting her work. We will follow up with attendees and provide the webinar along with contact information for both Claret and for Dr. Miller should you have any additional questions. Thank you.